Hi, this is Miss Litton, and this is the continual review. We are on Big Idea 2, considering it's Monday and your test is in a week. I don't know that we're going to get through the other big ideas. You might have to look at old reviews of big ideas in the past, of the ones that, start watching the other big ideas, because we won't probably get through all of them. So we're still on big idea number 2, but we are on slide number 4, Enduring Understanding, also 2B. So big idea 1 was about evolution. Big idea 2 is how we utilize energy to build energy and molecular building blocks to grow, to reproduce, and maintain homeostasis. And the part that we are on, 2B, growth. What words do you think of when you think of growth? What comes to mind?
Cellular growth.
Mitosis. Yeah, you might be thinking about meiosis and reproduction. You might think, when I think of growth, I might think of like the tropisms, right? Those all come to mind. Were those things you were thinking about? Okay, growth, reproduction, asexual, sexual, right? And homeostasis require that cells create and maintain an internal environment that is different from their external environment. What do you use? So you have a difference between an internal and an external environment. You have to have a cell membrane, right? That's what that's relating to. So if you're going to do all of these things, you've got to be able to maintain homeostasis. So then that brings us to the cell theory. Parts of the cell theory that all living things are composed of cells. Cells are the structural and functional units. And cells come only from pre-existing cells. We said cells are very, very what? Small, so that they have a large surface area per volume. Very good. We then went on to talk about the barrier, the cell membrane itself, and protein functions. There's some other things. I want us to look at this diagram to see what a cell membrane is made out of. A bilayer. What is that? Except for that one tricky question that you had on your practice AP exam where it was just a layer. Right? Not a bilayer. But phospholipid bilayer. And then what's the mosaic part of this fluid mosaic model? Proteins, and there are several different protein functions, and we'll review those, though I know you know them. What else is in here in our cell? Cholesterol. What does cholesterol help with? Fluidity. And then the inside and the outside look different. Amphipathic, right? They look different on either side, because what do we have on the outside? Yeah, go say it.
Extracellular matrix.
ECM, extracellular matrix, or glycocalyx, carbohydrate side chains, right? And then to the inside we have our what? Cytoskeleton, right? Cytoskeleton, good. So this is creating a barrier. You can have a barrier around a cell. Within a cell, you can have little compartmentalization generated by barriers as well, other cell membranes, if you are a what kind of cell? Eukaryotic cell. Okay, good job. So then we talked about how do you go get from one side of the membrane to the other side. You cannot cross, you shall not pass if you are what? Larger charged. So what are your options? You can go what? Through the phospholipid bilayer. Okay, you can use a channel or a carrier or endo or exocytosis, right? This one, active or passive? Passive. This one. If it's a carrier, it could be active or passive. What do we call them? A pump. Yeah, I'm working a pump. Okay, so a pump. Let's pump it up, spike. Okay, so through the phospholipid bilayer, that's always going to be passive, right? If it's going through, you're not going to control that. If you're using a channel, it could be passive, but if it's a carrier, it could be passive or active. Okay, and then how about endo and exocytosis? What's that going to be? Active every time, because you're moving a whole membrane, and in order to do that, you're moving a cytoskeleton. Okay, so taking a look at this diagram, I like it a lot because it shows you just exactly what we looked at. You feel good about that?
Yeah.
Great review diagram. Here's another one. You should be able to recognize everything on that one too. Are you okay with that?
Yes.
What's this one right here? This is showing you not drawn to scale, but water molecules, right? We talked about there's through a couple of ways. It can go through the phospholipid bilayer, or it could use a aquaporin. And because it's moving a lot faster than it should be able to. And then that makes us think about osmosis. I couldn't decide which diagram, so I just put about 1 billion diagrams in here, but you understand the big picture on that. Let's talk about the different ways that the protein functions. We'll just go over them. There could be what? Channel, carrier, cell recognition, receptor, enzymatic, and junctions. Now, not all of those have to do with crossing, but the membrane, while it's there, the proteins embedded in those membranes have those different functions, and we had examples for all of those. Okay, so channels and carriers. Sodium potassium pump. Now that you have learned about a neuron, right, you should be like, boom, shakalaka, right, on sodium potassium pump. 3 sodiums out and 2 potassiums in. Sodium potassium pump is responsible for the, not the action potential, resting potential, right, of the membrane, right? The resting potential, when a neuron's at rest, the outside is positive relative to the inside, which is negative. Why? 3 sodiums out for every 2 potassiums in. What? Those are both positive. How does that make any sense? Well, there's more positive charges outside than inside. Inside there are also what? Negatively charged ions, and also leaky potassium gates. So that sets up our resting potential, right? The action potential is due to what?
The sodium gates.
Yes, the sodium gates opening, right? Sodium gates opening, and you might reach the threshold, right? The threshold. I was like falling through a threshold. I don't know. He got it though. The threshold, and then once you've triggered the then sodium gates start flying open, and that quick reversal of charge is the action potential. Remember that, right? Now, there's the quick recovery and long-term recovery. The very quick recovery, so the neuron can immediately fire again, potassium gates. Because sodium just came in to make the inside positive, right? Right? Potassium goes back out to return it. In fact, so well right away, it actually creates, do you remember? Yeah, it overshoots it a little bit. Yeah, it puts so much in there, and that keeps the action potential going in just one direction. The long-term fix, if you keep firing off that neuron, all your sodium will be inside and all your fixing potassium will be outside. But the way the gates work, you've got to get the sodium back out and the potassium back in, and we use the
sodium potassium pump.
Yes, which is responsible for the of the neuron, right? 30% of our ATP, right, is dedicated to sodium potassium pump. So that's a big one. All right. Cell recognition. What do I think of for this one? The immune system, right? Immune system. Just pausing off task just because I know we're not going to get to this one. It's in the 4th big idea that I think I talk about it. The immune system. Remember, there is your innate immune response, right? Where you just recognize self from right. And so you have things. Who's part of the innate immune response? Let's say something gets across your skin, your barriers, your membranes, your low pH, and it's in your body. Who's responsible? Neutrophils. 70% neutrophils, right? And they are just saying something's bad there, I'm going to kill it. It's not specific, right? Not specific. It just kills anything that doesn't belong in the body. The specific immune response has 2 parts to it. What are the 2 parts? B and T cells, which are part of the cell-mediated and the antibody-mediated immune response. Remember B cells? What do B cells do? Generate antibodies. Get released. They don't have to be there. Those antibodies, remember, it's the plasma cells, the specific B cells, that to that particular antigen have been triggered and conferred with the T cell that also recognizes it. Do you remember that? And they release those antibodies into the plasma, right? And they can travel everywhere and get into the tissue fluid and attach and mark a pathogen for destruction. Or the base, because they're Y-shaped, the base of the Y could get in a mast cell and trigger the release of, which causes vasodilation, which can then cause inflammation. Yes? Okay, the T cells, remember MHC1s and MHC2s. MHC1 and you display an antigen, you are done. MHC2 and I will, because you're a what kind of cell? Presentation cell. And you're saying, look what I've killed. You've chopped off the head and you're carrying the head around. You're not infected with it, you're just showing it. Okay, so, okay, I won't say anything. I'm not saying his head. But head is gonna. Okay. I'm not saying him. All right. All right, good. That's good.
That's the one character I know.
Receptors can also be enzymatic or just receptors. They could trigger, allow a gate to open to allow a secondary messenger to come in when we think about hormones and cell signaling. There's just so much there we could talk about. All right. There's enzymatic, aquaporins. We did that. Peripheral. Okay, that's awesome. Let's keep going. Osmosis. Diffusion of water across a
Semi-permeable membrane.
water must flow from the hypo. Okay, so it's going. Osmosis is a great example of entropy, right? Increasing entropy, because you're going from water where it's more concentrated to where it's less. Yeah. So this side here on the right, if this is side B, and this is B, and this is side A, okay? Which side is hypotonic? Hypo. A. And side B is? Now the water would want to move the other direction, right? And if it can, it will. Does the sugar want to move into A?
No.
Really?
Oh, yeah.
Yeah, it sure does. The sugar, if it could, this sugar molecule would move, but it can't because it's too big because of these pores, right? So it's staying put. So this is hypertonic. Water is going to flow in there, right, to try to equalize that until the osmotic pressure, the force of gravity, pushes down and pushes as much water back as that's going in. Yes? Okay. Who has more osmotic potential, A or B? Potential. A. Who has more osmotic pressure? B. Okay, good. Water potential. Water potential is a factor of two things: pressure potential and solute potential, right? Solute potential is how many OASs you have, osmotically active substances in there, right? The hypertonic one, right? It's going to come in. Osmotic pressure has to do with the wall pushing back, right? Just like I told you, water is going to move into the sugary side until the pressure potential, right, is going to push back. Gravity is going to push back, the force of gravity, right? So in the same thing, the wall is going to push back. So even though you might have a very hypertonic cell, eventually the amount of water going in is getting shoved out due to the cell wall, right? So isotonic says same thing. Same amount of water that's going in is flowing out, okay? And hypotonic, that if this solution is hypotonic, the cell must be hypertonic. So it could go into a red blood cell and actually cause it to burst. A plant won't because it has a cell wall.
Pressure.
Yeah, and so the turgor pressure. That's why you talk about water potential with plants, because they have a cell wall that can push back. It's not just a factor of the osmotically active substances like it is here with our body cells. Okay, hypertonic solution, water must flow from the hypo. So this is water's coming out into this hypertonic solution. So it's shrinking down, and here you can see the water has left the large central vacuole, and the cell membrane is pulled away from the cell wall. You okay with that? All right. Oh, look, water potential. Turgor pressure due to the cell wall pushing back. Oh, I already went over this with you. Easy peasy, lemon squeezy. Oh, how do we feel about this? Actually, already went over this.
Yeah.
Okay. Facilitated transport. Just making sure I pushed play. I couldn't remember. Facilitated transport. If I'm facilitating, I'm just using a protein to facilitate a molecule going from a higher concentration to a. It's not costing me any what?
ATP.
No ATP. Good. When sodium's outside and it wants to come inside a neuron, when an action potential is going on, that would be facilitated transport. You can't it can't go through the phospholipid bilayer because, not because it's large, but because it's charged. So it has to use a protein to go in. It's very specific just to sodium. And when potassium resets the neuron quickly, it's using a protein. in order to do that. That gate is going to open and potassium can go back out. Specifically just potassium out that gate. And those gates tend to be what? Leaky. Okay. Active transport, we've already talked about that. Endoexocytosis, follow your arrows. If you're going in, right, if you're going out. One type of very specific endocytosis is called what? Receptor-mediated endocytosis. So it binds with the receptor and then they do endocytosis. Oh, and there it is. I'm just getting ahead of myself. I'm going to keep going. Endosymbiotic hypothesis. We already kind of talked about this. This is how a eukaryotic cell formed. What is our evidence for that? Mitochondria and chloroplasts could have been free-living prokaryotes, but what's our evidence for the endosymbiotic hypothesis? Double membrane, circular DNA, self-replicating, and the are the same size as a prokaryotic cell. All right. Hey, there's a eukaryotic cell. We know what everything does there. Hey, there's another one. Let's just go over these real quick. Nucleus. What is this nucleolus? Gene amplification, multiple copies of the DNA, right? And nuclear membrane, what's this going around it?
ER.
What kind of ER?
Rough
Rough
ER.
ER. What? Rough because what's on it? To facilitate what? Protein synthesis. And then the protein can move into the ER and then get transported to the smooth ER. Where might it go next? Where it will be processed, packaged, modified, either to be released or kept within the cell. You okay with that? Stomach of the cell?
Lysosomes.
Lysosomes. Good. Okay. Centrioles. Everybody got them? No.
No.
Who doesn't? Plants don't have them, right? Plants don't have them. Okay. Mitochondria? Powerhouse. Okay, good. Cell membrane, cell wall, chloroplast, thylakoid membranes. We see all of those. We're good on that. Vacuoles could be used for storage. They could be used for excretion. They could hold pigments. They're like the multi-purpose structure holding the cell upright. They are the multi-purpose room of a plant cell. Okay, they can be used for many different things. Okay, we know that. We know that. We know that. We just talked about leaving the rough ER and going into the smooth process package. Yay, we're on 2C. Woo! Okay. Organisms use feedback mechanisms to regulate growth and maintain homeostasis. You know, homeostasis, right? We talked about the different kinds of feedback that we would have. Negative feedback. Negative feedback, does that mean you're mean? No, you're trying to keep a balance, right? A set point. How could you do that? You could do that with hormones. Yes, what kind of hormones? They could be contrary, or what's another name for it? Antagonistic hormones, right? Could do that. You're trying to maintain a set point. We're good with that. Let's talk about this feedback, this negative feedback. Do you remember these hormones?
Yes.
Okay. Hypothalamus. Remember the pituitary gland. There's the anterior pituitary and the posterior pituitary. Remember that? Okay. The posterior pituitary, it's directly connected to, right, via the stalk. The anterior pituitary grew up and out of the mouth. So it can control, the hypothalamus can control the posterior pituitary with neurosecretory cells extending right into the posterior pituitary, both making the hormones that it's going to be using, what it's going to secrete, and then the posterior pituitary will store them until triggered for release. Yes? What are those hormones that the posterior pituitary stores for the hypothalamus? Oxytocin and antidiuretic hormone. I don't want to die. It's an antidiuretic hormone, affects collecting ducts of the nephrons, and it helps with water reabsorption out of them, right? So it goes back into your body at the nephron, yes?
Yes.
Okay, because I don't want to die, I got to reabsorb that water. Take the water out, yes? Okay, water can relate to oxytocin. Oxytocin is our out hormone. What two things come out? Milk and the baby. Okay, and you need water to make milk for the baby who's coming out, right? That's maybe how you can link them all together. The anterior pituitary is indirectly connected because it grew up out of the mouth and grabbed on. So in this case, we use not one set of capillaries, but two. Okay, so neurosecretory cells put releasing hormones, QRS, R comes before S, releasing hormones into the stalk, into the first set of capillaries. That goes in the blood to the anterior pituitary, and they come out and tell the anterior pituitary. They come out at the second set of capillaries, yes? And then the anterior pituitary can put hormones into that set of capillaries, and it gets brought back to the heart, to the lungs, to the heart, and to the body, yes? All right, now, a good example of this is when you do not have enough thyroid hormone, then your hypothalamus will secrete thyroid-releasing hormone, which will go into the first set of capillaries, which will travel to the second set of capillaries in the anterior pituitary and stimulate the anterior pituitary to secrete thyroid stimulating, QRS. See, R comes before S, stimulating hormones, which go out into your body, and they will stimulate the thyroid gland to secrete thyroid hormone. As the thyroid hormone level builds, it exerts what? Negative feedback. It says we have enough. And it shuts down the hypothalamus and the pituitary. What's a good example of when that doesn't happen? When somebody gets a goiter. Why? What's the problem with their goiterness besides not fitting his shirts well? You don't have enough iodine. How does that? No. Yes. Why? Yes. Why is that bad? What do you need iodine for? To make thyroid hormone. Yeah. So if you don't have enough iodine, you won't make thyroid hormone, so there's nothing exerting feedback. So the hypothalamus keeps stimulating the anterior pituitary keeps stimulating the thyroid gland, it keeps getting bigger and bigger and bigger. How do we solve that problem? Iodized salt. If you have enough thyroid hormone, then you can't exert negative feedback and it won't overstimulate. Question?
Oh, so it's saying you need iodine for TH, but if you don't have iodine, then it's not regulated. And that's
if you don't have iodine, you can't build the TH. No TH, no negative feedback. Everybody's still yelling at the thyroid gland.
Because it's overproducing.
Because it's not making any thyroid hormone.
But then why does it build?
Oh, it keeps getting bigger because it's stimulating the thyroid gland. It's thyroid stimulating hormone, so it's going, go thyroid.
But it's just not doing
yeah, and it's like, I'm trying, okay, but it can't. Okay, all right. Temperature, homeostasis, feedback on that. Unless you have an infection, and then your fever might get up higher because you're trying to, that's going to override it because you're trying to kill off that bacteria that's in your body. Positive feedback, would you like a cookie? Yes, I would like a cookie. Would you like a baby?
No.
No. Okay, no, but not. Okay, there's one way to solve that. Okay, not have sex. Okay, so what happens is the baby, just throwing it out there as an idea. Apparently, if you don't put a penis in a vagina, I'm so sorry, you get a lesson here. If you don't put a penis in a vagina, babies aren't made. I know, it's weird.
Are you saying Jane the Virgin though?
Yeah. Okay. There's one virgin, her name is Mary. Okay, now, come back to me. The dragon's out. Okay, the genie, whatever that. Okay, come back to me. Focus. Cervix gets pushed on by the baby's head. This makes your hypothalamus, make your posterior pituitary make even more oxytocin, which makes the uterus contract even more. Okay, so this goes on until you have some climactic event like you have the baby. That would be fairly climactic. Okay, ethylene and fruit ripening. That's another example of positive feedback. Remember we talked about how that's a gaseous hormone, so it gets released into the environment, which makes other fruit ripen, which makes other fruit ripen, which makes other fruit ripen.
Yeah, like the banana thing.
Yes. Here's one, another positive feedback loop we can put into global warming. So look, we talked about the body, we talked about fruits, and then we talked about global warming. So in global warming, in this case, a change in temperature is warm, so the ice melts. The ice is reflective. Now there's less ice because it's melting, which means even less reflective surfaces, so it gets even warmer, which makes more ice melt, which means it gets warmer, which makes more ice melt. Do you see the problem? And you reach this tipping point, and that's why we're losing all that old ice, right? And we just have this new ice. We don't have enough of that old ice forming, and we're going to reach a tipping point and, yes, move somewhere else. Okay, next. Positive feedback in your menstrual cycle, right? Because hypothalamus secretes gonadotropic releasing hormone. which makes your anterior pituitary secrete the gonadotropin FSH, follicle what? Stimulating hormone, which stimulates the follicle in your ovary, and the follicle in your ovary will start to secrete what? There's 2 hormones out of the ovary, and they are produced in alphabetical order. Estrogen. Estrogen causes 2 things. It causes the buildup of the uterine lining, and it gives positive feedback to the hypothalamus, which makes more GnRH. So there's more buildup until you reach that tipping point, just like glaciers melting. Okay? Reach that tipping point, boom, then that's going to trigger then what? Follicle stimulating hormone, which makes the egg burst out of the follicle. Yes, the follicle, it's follicle stimulate. Oh, did I just say corpus? Luteinizing hormone, which makes luteinizing hormone, which causes the ovulation. I don't know what I said back there. I'm trying to correct myself. So FSH makes the follicle mature. Follicle secretes estrogen. Estrogen does a positive feedback loop until you reach a tipping point. Then you're going to have luteinizing hormone come out and from your anterior pituitary. It causes the egg to burst out. That's called what when the egg bursts out? Ovulation. And it makes the follicle get turned into this yellow goopy mass called the corpus luteum, and now it secretes the second hormone, progesterone. Progesterone maintains the uterine lining and exerts negative feedback on your hypothalamus. If the egg gets fertilized, HGH hormone is generated, HCH, not human growth hormone, sorry. And as a result of that, then you're going to keep the uterine lining until progesterone can be made by the placenta. But if not, you will have no more progesterone made, and as a result of that, it'll start to wither away and you will get your period. Okay, so there's both positive and negative feedback in your period. Okay, and there's a picture. Here's another example. Okay, this is where you're trying to keep your blood sugar levels what? Just right. So you use contrary hormones like insulin and glucagon. There you go. Okay, we got that. We did that. Organisms respond to changes in their environment. What do we see right here? What's going on here? Yes, and because you're trying to test phototropism. Here is something in your environment, a stimuli, the nervous system. You detect that stimulus and you give a response. If you are outside and it starts to get cold, you detect that and you will what? Shiver. That's responding to your environment. Lizards will turn, make their bodies so that they can absorb the sun. They will do behaviors in order to, they do this actually, and they're trying to absorb as much light energy as they can. All of these are responses to your environment. Oh, look, we ran out of water, so we're just going to shrivel down and do anhydrobiosis and wait until we can become active again when water is there. Look, see the water bear? Up here. Oh, behavioral response to the environment. What do bears like to do? Hibernate. And why are they doing that? Probably because there's not a lot of food at that point. So to hibernate and just slow down their body systems is a great response to their environment until food becomes available again. Let's look at another. What's happening here? Migration. Why would organisms migrate? Follow food. Maybe there's a better place to mate than there is to raise your young. So it means that the environment is not always consistent. It might change. So warm summer months may be followed by inhospitable cold. So they want to migrate to a new location. Sometimes the journey's long, sometimes the journey's short. Have you seen all the butterflies migrating?
Yes.
It's crazy.
But it happened earlier, and it's happening.
And now it's happening again. Thermoregulation, we talked about this. I already mentioned this. Your body responding to whether it's hot, it sweats and dilates in your blood vessels, and your capillaries and towards your skin will dilate in order to cool you off, or you shiver and redirect the blood towards your core. Countercurrent exchange. Where have we looked at countercurrent exchange?
Fish.
Lungs, blood. Look, oh my gosh, we're almost done, and then we'll have done 2 of them. And here's another one. Mutate to deal with environmental stress. Now, between now and when I see you tomorrow, no mutations. Oh my gosh, too soon? I don't want you to be stressed out. So we're partway through. If you look at this, do you see where this is right here? That means how much presentation I have left in just big idea 2. That's why I'm saying.
Is big idea 3 smaller?
Yeah. I'd say it's about half as big in big idea 4, so you might just need to watch those independently because we're going to spend some more time on big idea 2. Plus, I'm also bringing in stuff that I know I'm not going to have time. I went into the immune system because I know we talk about that in big idea 4, so I'm just kind of bringing it in as we go. I'm trying to tie in as much as I possibly can. Yes?
If big idea 3 is half as big as this one, why don't you spend 3 days on this unit and 1 day on big idea 3?
We might be able to, yeah, I hope, if we can finish it up tomorrow. I can go a little bit later tomorrow because I have a meeting at 3:30. So if we want to go later and make sure big idea 3 is completely done. Big idea 4 has a lot to do with ecology and things like that we just finished up. So there's some of that in there too. Okay. So keep going, keep reviewing, and I will see you mañana. Make sure your hyperdoc's good to go the first part. Yes? Yes, play God Day tomorrow. Have a piece of toast, and we will start on 2D next time.
She's talking about the next